Thank you, everyone, for coming to this Friday's POC apps seminar. I'm going to introduce our speaker for today, Ailshire. That's about the only thing I'm going to say about Jennifer, because Nathan's going to talk about Jennifer. So I'm really happy that she's here.
So our speaker today is Jennifer Ailshire. Jennifer is a professor of gerontology, sociology, and spatial sciences at USC. She's also the co-director of the USC UCLA Center of Biodemography and Population Health. Jennifer's research focuses on how our social and physical environments affect our health and well-being throughout the life course, but especially in older ages. Today, I'm excited to hear about, we'll be talking about the exposome and how population health research can help us better understand it. Please join me in welcoming Jennifer.
Thank you, Nathan. I've had to introduce a lot of speakers and I know how much work it is and how stressful it is, so I appreciate that. Thank you so much for having me here. I have so many friends and good colleagues at UT Austin. I surprisingly don't actually collaborate with many people at UT Austin, but I sure do like these folks. And these are the people I find myself hanging out with when I see them at conferences. So coming here has been really great. It feels like being among friends and families. So as Nathan said, I'm going to talk to you about a new concept. And I'm doing this in particular because I actually think this is one of those things that sort of arrives on the scene seemingly out of nowhere because we're not aware of its evolution. We don't know what it is. We don't understand it. In this particular instance of the exposome, I think we would be doing ourselves a disservice to not get a better understanding of it. So I'm here with a very specific agenda. And this talk is very kind of biosocial in nature, which I know fits well with some of the focus areas that you have here, both in the Pop Center and in apps. So the exposome is really a new kind of paradigm for thinking about how we study not just how we study the impact of environment on health and aging, but actually how we understand aging. I don't use the term paradigm shift lightly, but I do think that this is kind of a modern example of like a Kuhnian paradigm. And so I'll explain to you why I think that. But just so you understand what is this exposome that a lot of people at NIA are talking about now. It is the totality of environmental exposures, first off. So it's thinking about social, physical, and chemical environment. These are all things that you all already study in some way, shape, or form. You can grab any one of these icons. I'm sure everyone in this room can point to something that they've been studying or that they want to study. But the second point on the exposome is it's really trying to understand what are the impact of all of these exposures on sort of biological health, age-related changes in response to the environment. And then the nice thing about the exposome, it's very kind of sociological in nature, is the acknowledgement that all of these exposures and impacts on our human biology are occurring over the life course. And so there might be life stage specific impacts or cumulative exposures and experiences that we need to think about. So I think you already are probably feeling pretty familiar with like social environmental exposures, life course, health. So the word might be new to your ears, but the concept it's tapping into is very familiar to you, I'm sure. So the reason I think it's a paradigm shift is that in the study of health, we've traditionally had two camps. There are the folks that are really focused on biology and genetics. They're very much studying the processes and structures that are internal to the body. And then there's people like us in this room. We're immersed in the social and physical world. We study all of the factors that are external to the body. For the most part, we don't interact. We don't really talk to each other. But with this new emphasis on the exposome, we have no choice but to interact a little bit more. So I'm going to talk about some of the work that I've been doing kind of at the intersection of these things. So this exposome approach, which is for population scientists, it feels quite new. It is actually not that new for biologists. So biologists have been evolving towards this exposome paradigm over several decades. And I just wanted to give you a few examples. So a really famous biologist just wrote a book saying that genes are not the blueprint for life. I don't think any of us would have ever thought there was a blueprint for life. There was a lot of excitement over the genome, though, and there was a lot of work that got invested, a lot of time and a lot of money, mapped the genome very successfully. And then we ended up explaining no more than like 13% of the variation in human disease. And so that was an understanding that the genome is important, but there are other things that are equally or perhaps even more important, or at least at a minimum, that the importance of the genome cannot be understood outside of this exposome, this complete total environment of lifetime of exposures. So the exposome is this new paradigm for environment and health. People talk about complementing the genome with an exposome. And it's really trying to get this marriage between our environmental focus and molecular focus on health and aging. So just to give you a little bit more background, I started graduate school in 2003. That was a really good time for people who were interested in neighborhood health research. That was the group that I was working in. But neighborhood research, if I just did kind of a basic search, I looked for like neighborhoods or environment and aging, because when I started in neighborhoods, most of the work had been done on children and adults. Very little of it had been focused on older adults. Although there were some like original pioneers in the field like Powell Lawton, who had done a lot of work on aging in the environment, but somehow where he left off, it didn't quite ever get picked up again. And so we had this bit of a gap. But then there was just been an explosion of interest in understanding how the environment impacts our aging experience and how older adults are living in their external world. So there's a lot of focus, for instance, on constructing age-friendly cities, but in order to do that, we need a basic science research understanding of what a age-friendly city is. So the WHO has done a lot of work trying to understand that. And then most recently, there were researchers that were really trying to draw attention to the potential for an Alzheimer's disease exposome. We're in a moment in the aging world where we have improved a lot of health outcomes. We have this other health situation, which is Alzheimer's disease and related dementias. A lot of people are working across a lot of different fields to try to understand how to tackle this new challenge of having a lot of people who now live to very old age. And when they survive all of the historically traditional causes of death, they've just survived into higher risk for something like dementia. So actually, one of my colleagues who was a biologist would talk to another colleague, Eileen Crimmins, and I for years about the exposome. And we kind of laughed about it. But it turned out he was talking about something that was going on in the biology area, and he was pushing an agenda at NIA. So they had this workshop back in 2020 on the exposome and brain aging, Alzheimer's disease, and related dementias. This is where the new focus on the exposome coalesced and moved into our world. So the biologists were talking about it for a much longer time than we've been talking about it. And so it does come from, I think it's interesting, it comes from the field of biology and understanding that more, they need to move more towards the work that we do. And this is a huge paradigm shift, because for the longest time, we really thought of like biology here, social sciences way over here, and never the two shall meet. And now we're at a moment where we're going to be working together a lot more. So what's the role of population health researchers in advancing the study of the exposome? I think that we are the ones who have the data on life course experiences and exposures. We also have the analytic tools. And we have, most importantly, the theories and conceptual frameworks that really think through what an exposome impact on health might look like. So I'm going to talk to you about, some of the opportunities here, some of the work that I've been doing with my group, and also some of the challenges. And also just want to point out that the NIA is really investing a lot of research resources, a lot of effort into building up infrastructure to support population researchers. So one of the ways they do that is by funding P30 demography and economics of aging centers, which we have here at UT Austin. I work at one at USC. They've also just funded a couple exposome coordinating centers. So they're pushing this agenda. This is the new paradigm, but they are putting a lot of support behind it. And so NIA really believes that it is the future direction of aging research for population health scientists. Okay, so if I think about neighborhood environmental exposures on health and aging, I've put this little kind of circle here. These are a couple of different buckets of types of contextual or environmental exposure. So you have the physical and built environment, the social environment, amenities environment, which is kind of where I think about like healthcare resources, and then the chemical environment. We all know about this idea of thinking about upstream to downstream factors. And so this is a very long known model in aging research about the process that goes from first disease to disability to mortality. People here at UT in the Pop Center have been pioneers in research on disease, disability, and mortality. What we're now trying to do is just back up a little bit to biological aging. And then I also want to at least acknowledge that there are these psychosocial and behavioral pathways. I think that's another place where we're going to be seeing more research. It's a great place to invest your time and effort if these are the kinds of topics you're very interested in. Today, I'm mostly going to focus on trying to unpack what this biological aging may be. Raise your hand if you've heard of an epigenetic clock. Raise your hand if you know what an epigenetic clock is. Okay. For those not in the audience, there were maybe half the people who said they knew what it was, but this is like a new kind of buzzy word we're hearing all over the place. And I just want to give a little bit of a background because I do think it's important if we're going to talk about these new measures of biological aging that we all understand what we're talking about here. So these epigenetic clocks or epigenetic age or accelerated aging are designed to be analogous in concept, at least, to biological age. And so they're constructed with that in mind. These clocks, the research that's been done to construct and then validate the clocks have shown that they do a pretty good job in terms of predicting health-related outcomes. So both the levels and patterns of change in the clocks are very predictive of age-related outcomes. That's kind of like an external validation exercise for these clocks. And a lot of research is coming out showing that they're related to adverse life events, improvements in the social environment, stress, environmental hazards. But like, what is a clock? So another big kind of buzzy word at NIA, not so much for the social scientists, but for the biologists and those of us who work in a field called neuroscience, is the hallmarks of aging. So these are all thought to be the molecular, cellular, and physiological changes that occur that are related to aging. And so it can be like, you know, mitochondrial dysfunction, cells dying, stem cells not getting regenerated, bad communication between cells. They're not talking to each other the way they should be. Some of you may have heard of telomere life. Telomere dysfunction is one of the hallmarks of aging. But epigenetic modifications are considered a major hallmark of aging. And I'm just going to give you the most simplified way I can think of explaining this, which is that epigenetic clocks are trained using data on DNA methylation. That in our population studies comes from blood, but this can be done in many different tissues. We usually only collect blood from individuals. And this is, I thought, a nice kind of teaching tool from a paper, but it's showing these CPG sites that are on in our DNA. And these are the CPG sites on top for young people, on the bottom for old people. These blue ones that are filled in circles, this is methylated, and the white empty circles are not methylated. So you can see that this older person, let's say, has more methylation at these CPG sites, whatever they are, and that they would have reduced gene expression compared to normal expression. The reason why the social sciences got pretty excited about the epigenome is because our genetics, our DNA is fixed at birth. And then the idea of using that to predict a bunch of things that happen later in life always seemed a little limiting for us social scientists because we knew an entire lifetime of exposures was going to happen after that. And also that genetic risk is real, but it depends on a lot on your environmental conditions. The nice thing about looking at the epigenome is this is the part of our genome that's responsive to the environment. So it's a great measure for social scientists. And so like if you have more methylation, say like in this older model here than in the younger one thing that could happen, for instance, that reduced expression, that could be methylation showing that there's reduced expression of a gene, of a tumor suppressing gene in cancer cells. That would be a bad thing. That means if you're not tumor suppressing in those cells, you might end up with more tumors, you might end up with cancer, and that's part of an aging process. Whereas normal expression could be the gene suppressor, the tumor suppressor cells turned on in the gene, it's functioning appropriately. So that's like one example of what this means when we say that if there's lots of methylation, that's bad. It suggests that you're aging at a more accelerated pace. I gave you a pretty clear example on tumor suppressing genes. This is actually a big black box that a lot of people are working on trying to figure out what all of these different CPG sites are related to. They could be related to gene expression. They could be related to anything going on in here. They could be related to cell signaling. So a lot of people in this field are working on this. I'm not working on it. I'm using the measures because my interest is in the neighborhood and environment. All right, so moving on, I just want to show a couple of things that I and my team have been working on to show you some of the applications, and the first is in the social environment. I started my graduate career working on the Chicago Community Adult Health Survey, and this was in the kind of Chicago School of Urban Sociology style work that Rob Sampson had been doing in the project on the PhDCN, which was a study of children in Chicago neighborhoods. And so I worked a lot on neighborhood disorder. For those of you who don't know, neighborhood disorder is sort of like what you see in these pictures. These are the signs of neglect and disinvestment, lack of safety in communities. That research has shown can be a pretty significant source of psychological stress among residents. And so there's been a lot of research showing that people who live in highly disordered neighborhoods might be experiencing heightened states of distress or heightened vigilance because the place is not safe. So they're kind of like always turned on. And this is why it's thought of as a stressor, external environment. A lot of research has been done on this showing that older adults who live in these kinds of environments have worse physical and mental health. So they have worse physical functioning, higher rates of depression, worse cognitive functioning. But another social environmental characteristic that's really important is social cohesion. This is generally thought of as a sense of connectedness and trust among neighbors in a neighborhood. It also taps into things like reciprocal exchange. Do you think that you can rely on your neighbors when you need help to help you with something? And this lack of social cohesion that occurs in some neighborhoods, we think of it as reflecting kind of a breakdown in that social contract or the social fabric of the neighborhood. And so it can result in low levels of trust and increased vulnerability, and people don't feel that kind of support. So this is sort of like network-based researchers would think of this in terms of your network extending out into your neighborhoods. And then people who work on social support would think about this in terms of social support. Most of the social support we think of is with family and friends, but neighbors can also be a source of social support. So one of the studies that I've been doing with a postdoc is looking at neighborhood social environment and epigenetic age. For that, we use the health and retirement study. In 2016, the HRS collected whole blood, and from there, they were able to get an increased number and diversity in terms of their panel of biomarkers. So this is about 3,000 people who are 56 and older who are in this DNA methylation subsample that was used to create the epigenetic clocks. For disorder, in the HRS, we actually have two sources of disorder. They ask the respondents to report on disorder in their neighborhood. But also, whenever the interviewers are there for a face-to-face interview, which they are for all the people they get blood from, the interviewers report on signs of disorder in the neighborhood as well. And then cohesion has to be reported by the respondents in this survey. The outcome in this paper is the epigenetic clocks, which I described in general, the sort of scientific understanding behind it. But when we make these measures, what we're actually trying to understand is, with respect to someone's chronological age, where are they in terms of their epigenetic or biological age? The idea is that this dotted line here is like a perfect correlation. If you're 40 chronologically, you look 40 biologically. But for people who have a biological age or the epigenetic clock age that's higher than their chronological age, we consider that here, the orange line, that's considered accelerated aging. So those people are kind of biologically, physiologically looking like they're aging faster than they should be based on, you know, everyone else in that age range. On the other hand, some people are aging more slowly. That's represented by the bottom blue line, because at any chronological age here, their biological age is actually a little bit. Everyone seems to be interested in their epigenetic clock, and if they're accelerated agers. I saw that the Kardashians just got their epigenetic age. I noted that Chloe, I think, at the lowest epigenetic age of the sisters, so a competitive sibling group. So this was another competition, but this is, I think, the science needs to be better done in this area, but this is one of the times when our work has fully jumped out of academia and into the mainstream. So it's, you know, it's a good idea for us to understand like what people are talking about. But that is the general idea, that you're either kind of younger or older than you should be based on your chronological age. Okay, and then these are the results. I have the full results here, and then I'm going to show you a very simplified graphic in case you glaze over when you see lots of these kinds of, because it's a lot. But we looked at socioeconomic deprivation, which we measured using census data linked at the census tract level. We also, as I said, had observed disorder, the interviewers who were in the neighborhood, the perceived disorder, and here this is reverse coded as low social cohesion. Those are both coming from respondent reports. You might also notice here, we actually have 5 different clocks. So these are all the clocks that got produced for users of the health and retirement study. I will say I do think that the recommendation for best practices here is to always present all of the clocks and not pick and choose, because in order for us to kind of make comparisons across different studies and like review the literature, it's a good idea to have all the clocks. You could put some of them in the appendix if you want. But so we're reporting all the 5 clocks here. So if I just start with socioeconomic deprivation here in the top left, we have a couple of different models. We start with model 1 that has basic demographic controls. We add socioeconomic characteristics like individual education, household income, and wealth. And then in model 3, we add some health behaviors. It's particularly important. We add smoking and BMI, because we know that those things can kind of determine where you are in your epigenetic aging. And then we also adjust for these cell types, which is like a kind of a scientific principle, but basically we don't want someone's epigenetic age to be reflecting the variation that people have in these that they provided. So you can see that these 2 clocks that are called Horvath and Hannum clocks, there's not really any relationship between neighborhood socioeconomic deprivation and aging. But we do see relationships with these last clocks. Although one covariate controls, we only see the final clock called the Dunedin pace of aging clock, that there is a significant association. This suggests that individuals living in socioeconomically deprived neighborhoods are aging faster, biologically speaking. Moving over to observed disorder, we find kind of a similar pattern where these first 2 clocks, there's no association. We find associations with the other 3 clocks. A lot of it's being explained by the kind of factors we tend to find would explain neighborhood associations with health, but still kind of seeing something here with that last clock, that an aging clock. We noticed that perceived disorder had kind of, you know, slightly stronger associations, or if not stronger, at least it was a little less noisy, because these confidence intervals were, you know, not as wide as they were for observed disorder. But again, kind of seeing signs that if you live in a more disordered neighborhood, you're aging more rapidly. And then here, if you live in a less socially cohesive neighborhood, you're aging rapidly. And so this is basically just plotting one of those, showing that you have aging acceleration with neighborhoods that have higher neighborhood disorder, and you have maybe aging deceleration in neighborhoods that are more cohesive, that have more trust among neighbors. Going back to this, I think a lot of people, when they do this work, it is incumbent on us as social scientists to then explain, like, why are we seeing relationships with some clocks and not others? So I do want to just point out those first two clocks that aren't showing a lot of relationships, or they're considered the sort of first attempts at clocks, first generation. They were trained mostly on age. The clocks that came after that were trained on multi-system indicators of multi-system physiological functioning and dysregulation. And so it may be that these later developed clocks are actually picking up on more of like gene that's relevant for thinking about neighborhood and environmental exposures. I don't know if you guys normally do questions in these seminars, but any one of you can feel like you can interrupt me at any point. Oh, so that was the social environment. I want to move on to some work we're doing in the physical environment. There's a lot of attention on heat. A lot of us around the country and around the world, particularly people who live in the southern United States and people who live in the global south, are experiencing these extreme heat events. Heat is really problematic for primarily two groups in the population: older adults, also younger children, and it has to do with some of the physiology around the way that human bodies regulate internal and external temperatures. So heat for older adults has been associated with increased risk of several different diseases like respiratory heart and kidney disease. It's also been extreme heat events have been associated with mortality. We saw really good work on this exact topic in Eric Kleinenberg's dissertation on the 1995 heat wave. That was a biological susceptibility, but it was also a social vulnerability because those older adults were socially isolated and no one was checking on them, and that's why they were more likely to die during that event. This graphic is just to point out that heat is something that's impacting the entire system. So this is, to me, why it's important to think about then measures that are kind of tapping into the entirety of aging rather than necessarily one specific disease. So if we think about impacts of heat on older adults, the ability for us to withstand the physiological impacts of heat is really variable by age. Most of us in here are in a pretty good shape. Like we'll do okay if it's a hot day. But if you're talking about young children or older adults, because of some physiology differences, they don't process that heat in the same way. So like older adults, even skin changes, the way that evaporative processes work change. But it is really important. Our body is constantly trying to move heat out of our core, keep it away from our brain. And when that's not happening, heat stroke, overheating occur, and that can result in negative events in both the brain and the body. So we're doing some work on heat and cognitive functioning and dementia, but that stuff's more in the works. I'm going to show you some work that's really far along, that's under review. Looking at those biological clocks. So again, we're using that same DNA methylation subsample, but for this one, we've linked some data on weather. What we've done is used something that comes from the National Weather System. It's this heat index. And so it's basically, it's getting people not just in terms of their temperature, but their humidity. And that's because you all live in Texas, you know the difference between a dry heat day and a humid heat day. That humidity definitely hits differently. And so it's important to kind of consider the two of them. I think it's also really important when we're thinking about older adults who don't have the same evaporative abilities. If that kind of reaction is impaired, plus it's humid, then one's ability to cool themselves is really impacted. So we create these categories based on the recommendations. So there's extreme danger, extreme caution, and caution. I'm sure a lot of you are looking like, if we're like 95 degrees here in Austin, what's the humidity? But I have spent most of my time working on air pollution, and we also have air quality indicators that people are now paying a lot of attention to. At USC, we had a heat wave a few weeks ago and fires. Our health office sent out advisories both for heat and air quality, and it was the first time I'd ever seen them do that. But I do tell people you should check your AQI and see if that's a good day to be out doing a lot of strenuous physical activity, or maybe better to stay inside the house. Okay, so I'm just going to show you 3 clocks because I think that like all the clocks start to get kind of overwhelming. But again, we do all the clocks in the papers for comparison purposes. So what we're doing here is we're looking at the heat index, that heat plus humidity on the blood collection day, 7 days prior, in the past month, 2 months, a year prior, 6 years prior, because we're also trying to understand like what's the exposure window that's really important here. And if we look at like this Horvath clock here on the far left, you can see it's really the more recent exposures that seem to be associated with accelerated aging. But the longer term, kind of longer cumulative exposure, we're not seeing relation. And this is showing it, we're showing here on this for caution and extreme caution levels. If we look at pheno age, all of the time frames matter. And then if we look at the Dunedin pace, only the cumulative timeframes matter. So I work on this work with an excellent early career researcher, Eun-Young Chae, and she is a psychologist, gerontologist by training. This is new to her. I just want you to know that like a year ago, she never worked on epigenetic anything. She did such a good job in this paper that we worked on, trying to understand why we're seeing different patterns across the clocks. I think she did a better job than I did. I think a lot of times early career people are more brilliant than we are. Maybe you don't think so, but you are. And so she and I looked at experimental literature, some of the more biology, animal-based literature, and we saw things that actually made sense because it turns out like in the animal models, the Horvath and Hannum clocks are really more associated with kind of immune functioning, immune activation, and we know that that's actually associated with heat as well. Whereas pheno age is very much a multi-systems clock, and so it made sense that all exposures, picking up on all exposures here, because this clock is doing a better job kind of getting at the whole of the physiology of an older adult. And then the pace of aging clock is actually trained on change over time. And so this one maybe makes more sense because we're looking at longer time intervals. I think for social scientists, biology freaks us out a little bit. It's not what we're trained in. You don't have to be trained in it. You don't have to be an expert in it. But I do think it's incumbent on us to try to understand when we link it with exposures, with social adversities, with social relationships, and we get these kinds of patterns, we do need to at least kind of dip our toes into the biology literature a little bit to try to understand what's going on. And then I always try to break these down into like easy to read graphs because, you know, even if you're a numbers person, we all have a day where we're just tired. So this is just showing just for the pheno age clock, 1 month and 1 year. And it's just showing that you know, caution is associated with accelerated aging, a caution level, but extreme caution even more so, and danger is really associated with that accelerated aging, but much more strongly in that 1 month interval than over the year. Again, we don't actually understand the epigenetic aging measures that much, and what we've been trying to understand is how much does that epigenetic signature pick up on your life course of exposures, and how much of it is being impacted on something you were just exposed to. So this is another area that we're gonna try to do. All right, so now I want to move into the chemical environment. This was a lot of the work I did after I left my PhD and during my postdoc. I was really interested in air pollution. Some of my sociology colleagues would ask me, what does this have to do with sociology? I just want to remind people that much of our physical and chemical world is the result of human activities, human choices, and it impacts humans. And it impacts humans differentially. So if you care about inequality, you should care about the physical environment because we've constructed much of the physical environment from inequality, and it is now reinforcing inequality. So I just want to put that caveat there. But now we're going to go really far into environment and health. But I do it all as a sociologist. So just to give you a sense of what I'm talking about here, I'm going to be talking about work I've done on fine particulate matter air pollution, which is one of the 5 criteria air pollutants that the EPA monitors and that we have standards and thresholds for. I also want to say that our environmental policy in the United States has been some of our most successful health policy. So the EPA started in like the 1960s. Its powers got reified into law in the 1970s with different iterations of the Clean Air Act. And it has been wildly successful at lowering levels of pollution in the United States. Obviously, we still have a lot of work to do. There's a new focus and there's a new bipartisan infrastructure plan that's focused on things like getting lead out of pipes, which we know has been a problem, and particularly in some parts of the country. So I just want to put that plug out there because I think for those of us who study population health, health disparities, and health inequalities, it's a little depressing sometimes, and it feels like overwhelming how much inequality there is. But there's good stuff happening, and people's lives are getting better. You know, some things are successful, so we have some successes somewhere. So I studied PM 2.5, which is 2.5 microns in diameter here. That's a pink ball. This is the diameter of a human strand of hair. So you can see how tiny these particles are. Or like this is a grain of fine beach sand. These are really, 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 really tiny particles. They're ubiquitous in the air we breathe. They come from industrial sources, traffic, and not just from exhaust pipes. So electric cars won't solve our problems with particulate matter because literally where the rubber meets the road, that's a grinding that happens. All of that turns into particulates. And also every time we brake the brake pads, that also produces particulates, and those particulates have metal. So older adults are particularly vulnerable to air pollution, especially older adults who already have some kind of respiratory or cardiovascular disease. When I started doing this work, there wasn't a lot of focus on older adults. Most of the focus was on children and adults. Since then, a lot more research has been done on older adults. This is the standard now at the EPA. It's 9.0 micrograms per cubic meter. When I started doing this work, it was 15. The science said it should be 12, but EPA, from political strategy or I don't know what, went with 15. They eventually revised it down to 12, and now it's at 9. But as someone who studies aging in older adults, I want to point out there's no level that's safe for human populations, and definitely not older adults. This is again just showing you that air pollution is something that impacts multiple systems. My focus is on the brain. And this is like the scariest photo I ever saw, especially because I just moved to Los Angeles and would sit waiting for the bus. But particulate matter is so small, it can actually transverse this olfactory bulb and get direct. So like the people who are saying like, how do these exposures get under the skin? That this is one of the ways that air pollution does. It's because it's so tiny. It's related to a bunch of different health outcomes. Okay, so this is work that I did back in my postdoc, which seems like not that long ago, but I guess it was now. Just showing the relationship between concentrations of tract level air pollution, 5 concentrations where people lived in health and retirement study. And I just had kind of quartiled it. But you can see actually like this first quartile is now right about where the new EPA standard is. But basically this is a score on a cognitive assessment, and these 2 lower quartiles, folks who lived here in the kind of lower pollution environments, had higher scores than the people who lived in these 2 higher quartiles. I know this one looks like it goes up, but these are actually not statistically significant. It's just a little bit of noise in the data. But because I'm a social scientist, a sociologist, and a demographer, I know there's a lot of population heterogeneity, and I was just convinced that like not everyone can be suffering impacts of air pollution, or there'd be like everyone in Shanghai and Beijing and China and different cities in India with lots of pollution, or Mexico City would all have problems. And because I was coming at this from a neighborhood social environment perspective, I was really interested in the synergistic association between physical and social environment. So the brain is considered the target organ for stress because of some things that are happening in the structures of the brain. Chronic stress has been shown in research to be associated with damage to our neurons and decreased performance on cognitive tasks. And then so I thought, well, maybe neighborhood stressors would be like a modifier of that relationship between air pollution and cognitive functioning. Again, there was not really a lot of research in older adults, but I did find some very good studies in children with kind of good conceptual framework. I just lifted that into older adults. But they had shown that children who were exposed to a lot of stress in the home or school or neighborhood environment, also exposed to air pollution, fared worse on their cognitive exams. And so I just kind of thought about this little model that if we're interested in environmental hazards and health, in this case, I was interested in cognition, whether or not it would be exacerbated by the presence of some kind of social environmental stressor, because it would operate through that psychological stress pathway, increase some biological susceptibility. But really, it was this relationship up here that I was examining in this paper. So this is the data again, just showing that with increased concentrations of PM 2.5, cognitive performance declines. And this happened, doing this in the health and retirement study as well. But we also looked at this, categorizing these respondents into groups, depending on if they were in a low social disorder neighborhood or a high social disorder neighborhood. Using all of those characteristics I showed you before, there's like presence of trash, deteriorated, broken down buildings, broken like that. And so what we actually found was that in the low disorder neighborhoods, there was no association between particulate matter and cognitive functioning, and it was all happening in the high disorder neighborhoods. And so, you know, like this really makes us think about like the total environment, trying to think about, again, about the totality of the environment, which is one of the definitions I gave you for the exposome. So I'm going to move now into just kind of some of the new opportunities or challenges, depending on your perspective on things. One is trying to get incorporating the timing and duration of exposure. So I think of this in two ways. You could have exposure happening in critical periods. So we know, for instance, that children, if you're exposed to lead as a child, it actually gets stored in the bone. And during bone remodeling processes that happen with aging, also during pregnancy, that lead can actually get recirculated back into the bloodstream. So that's an example of an exposure period approach. But we also think about exposure time. Like, have you been exposed to this bad thing just once and you can recover maybe? Or are you being exposed to this bad thing repeatedly? For surveys of older adults, we have a bit of an issue, which is we haven't seen them over their entire life course and we don't have data on their life course. And so people always ask me about doing a life course study and I just like don't have the data for it, right? So I'm showing you data from the health and retirement study, just to give you an idea. You know, this is one of our biggest studies of aging, but actually we can trace some of these cohorts back pretty far. So this early Gen X cohort's just been released in the new HRS early release data file. We also have these 3 baby boom cohorts here. And just taking something like the census starting in the 1970s, like we have censuses going back, but this is what I think of as like a good census year to start with, because the data are a little more complete. The race ethnicity categories make a little more sense. For some of these cohorts, like the newest ones, we're going to pick them up around when they're age 10. So we're going to see them for a good chunk of their life course based on data that we can link starting in the 1970s. But even these baby boomer cohorts, if we could figure out where they were in their 30s, we could get a good chunk of their adult life course at least. So you would have to get historical data, but we do have some of that data that could be linked. And I think the subsequent cohorts, so they've done early Gen X, they'll do their middle and late Gen X. These people are going to have information since, we're going to be able to link information since birth, which I think is really exciting. It's something we haven't been able to do in the United States. Other countries have a lot of these birth cohort studies. And then we asked people where they lived throughout their lifetimes in the health and retirement study. We're trying to code these right now. I can tell you it's a little bit challenging. So, but it's underway. So we're trying different things in HRS to try to get that linkage going far back. So very impressive and creative people like yourselves can do this life course of exposome exposures. I did want to point out just one paper that's already done something really interesting. It was done by Jean Choi and Liz Munoz, and I think Say, who's also in here. They actually used the life history part of the HRS to ask people about their perceptions of their neighborhood cohesion at different ages. And I made these figures. I hope they're okay. If you want, I'm happy to send them to you. Because, you know, it's like a paper with a lot of tables and numbers in the table, but I like figures. I'm a visual person. But it's basically showing that neighborhood cohesion at age 10 and then their current level. were both associated with levels of cognitive functioning, not so much with decline, but we don't find a lot of things associated with the rate of improvement, with the level. I thought this was a really creative way of thinking about a life course of exposure, in this case focused on social cohesion, because that's the data that they could find, but hopefully when we have more data for people, they can do more things. So if you wanted to do research on the exposome, where would you get these data? So I developed a contextual data resource that has different kinds of measures in the physical and built environment, the social environment, amenities environment, and the chemical environment. These are all available on our webpage. The link is up there, but it's a contextual data resource available through the Biodemography Center. I'm going to go through kind of quickly because I'm running out of time, but there's a bunch of different data sets that are available. We have them available at different intervals of time. But some of them you can see go back to like the 1979 is the latest we have for weather. And then we have them also available at different geographic levels, which is a whole other talk. So these data are currently available for people who want to use them with the health and retirement study. If you get a restricted data application approved, you can use these in their virtual data infrastructure. We also link them to the Understanding America study, which is a study of adults. It's not just older adults, and they use a high frequency design. So they're talking to people basically every month. And then if you work with the NHATS or the PSID, these data are also put into the geographic linkage repository. Okay, so I just told you how important the exposome was. And there's this sort of Garden of Eden awaiting us, but like, how do we get from where we are to where we want to be? I think that there are a couple of challenges. We need multidisciplinary teams. We can't all be experts in everything. We shouldn't even try. But we need varied expertise for these really complex questions that we have. Centers like these, I think, are a great place to start thinking about multidisciplinarity. I know like CAPS has got sociologists working with neuroscience people, and those are connections I think that are hard to forge and wouldn't be forged without center infrastructure. So that you always have us at the Biodemography Center. We basically have like a help desk approach where people email us and we answer questions. My colleague Steve Cole answers questions on lab science type things in biomarker collection and assays, answer questions on the data resource. But I think one of the other challenges, which is like it's like good news, bad news, is we're about to have an enormous amount of data. It's increasing, I think, at an exponential pace. I've shown you one set of products I've made. I could tell you about a half a dozen other places you could go to get a lot of contextual data. And so I'm really trying to understand, like, how are we going to model all these multiple exposures and over a very large part of the life course? We don't have those tools yet, although every time we've needed tools like that, the field's done a good job making them. So I'm optimistic. And then I also want to point out, since I assume there might be many PRC people here, that for demographers, we think a lot about population heterogeneity. One question I think that really needs more attention is whether or not the measures we have on both of the exposure side and the biology side are working for everyone. Those clocks that I showed you were trained in the health and retirement study. The Dunedin PACE is trained in a group of people in New Zealand. They're trained on European ancestry populations, and we're not totally sure how well they work in other populations or if we need to train different clocks for different groups. And I also think we're going to have to think about modeling approaches that account for the within population variation in some of these relationships. And just to show you one example of something I worked on with a colleague who works more in education than I do, we were trying to understand how PM 2.5 was related to incident cognitive impairment or dementia. And just to quickly show you, we only found the association on this line. This is people who have very low education in the health and retirement study. These are older adults who didn't make it out of primary school. So that's some heterogeneity that suggests to me that we would make a type 1 error, basically, if we said, yep, exposure to particulate matter is bad for your brain. But maybe it's only bad for the brains of people who lack the kind of resilience and resource cognitive reserve that you get through education. Flip side of this is a type 2 error. Me and my team spent so long doing an analysis looking at PM 2.5 and epigenetic clocks. And I've never had so much null findings in my life. It was like the nullest of null findings. I mean, this is zero, it's nothing. But I've been working with someone who works on machine learning. It's a neuroscience PhD student, but he works in computer science. And he created this hybrid regression guided neural network approach, this model. We put in a bunch of characteristics that neural network model basically goes in and tries all of the multi-way interactions that we can't get into our regression model, and then it produces a vulnerability score. It's really similar to the epigenetic score, actually. And then once we stratified based on this vulnerability score, what we found is that the least vulnerable, or the most vulnerable, so the top 10% on this vulnerability score, see that there was an association between PM 2.5 and their epigenetic aging. So this is very new work, but it's a direction we're going in that I'm excited about as a population scientist and a demographically trained one, because this issue of population heterogeneity has plagued our field for such a long time. And if there's a new tool or trick that we could use, I think that would be great. All right, so I just want to conclude by making 3 points. The first one is that this integration of biology and environment, and I study neighborhood environment, but we're talking family environment, school environment, your social world. This is where we're going in aging. So shouldn't be wary or afraid of it. I actually think you should embrace it, because you're more prepared for this seemingly new direction than you think you are. Because I actually think the exposome was a new word, but once I understood what it was, I realized this is what we do. We have a lot to offer to this new direction. And there's so many challenges, but I just think there's also incredible opportunities here. People can build entire careers helping us figure out, all of these interesting questions that are going to be so important for improving population health and improving the aging experience in the United States and in other countries globally. And then I also just want to point out, this kind of work always takes a village. I've been incredibly fortunate to have a lot of funding support from NIA, but also I've had the best postdocs and pre-docs, and working with mentees has been one of the most delightful experiences of my career. And these people, they're the people that keep me excited and keep me getting up in the morning and heading back into work.
But we seem to have questions come from graduate students. And we have 1 or 2 graduate students here at the so graduate students, you can raise your hands. There are no graduate students.
Hi, thank you so much. This was a really amazing talk. I'm really interested in thinking about the intersection between individual level and neighborhood level. So for example, socioeconomic status is often controlled for to isolate that environmental exposure. But looking at some of this work with education moderation, I'm curious, what are your thoughts on how do we account for that intersection between individual and neighborhood kind of variables?
I think that we still have a lot of work we can do there that would be really helpful. Understand that so different people live in different neighborhoods, but even within a neighborhood, people will be differentially impacted by the exposure based on their personal and family resources. And so trying to understand something like that interaction between your individual level resources and a more macro level context, I think would be really useful. I don't think a lot of people have done it. You have a data need. You do need to have enough variability, right? You need to have people who say have low individual SES and high individual SES living in similar kinds of neighborhoods. Otherwise, you're sort of outside of what your model can do. But there are many data sets now that have really large sample sizes. They've sampled in ways that I think brought a lot of that population heterogeneity. And so these samples have a lot more heterogeneity than we think they do. But that kind of a question, I think, is sort of data hungry, because you need to have a lot of data in order to look at the interactions. I will say, when I was looking at the pollution exposure and neighborhood disorder, one of my first questions, and the question I always got when I presented the work, was, isn't it just that people in high disordered neighborhoods those are also the highly polluted neighborhoods? So I had to do a good job kind of checking that out to see that actually there was a lot of off diagonal representation, and so it was a question that there were data to provide an answer for. But I think you're on the right path, and that would be a great direction to go in, like individual SES, but also other kinds of resources, like interpersonal resources, your family resources. Living in a stressful neighborhood is hard, but if you go home to a strong support network, I think that helps a lot in stress research in general. Yeah, I should also say I'm not even a stress researcher, but I read a lot of the kind of like psych work done on stress because it makes sense to me as a neighborhood researcher to think about, you know, what's going on with these neighborhood stressors. But yeah, I think that's a great set of things.
Education project, anyone? Another question from the graduate students?
Hi there. Given that HRS is obviously a sample of older adults, how have you thought about survivorship bias? Like, I'm thinking about how it's possible that people have the largest impact of environmental exposure might not end up in the HRS sample. And I'm wondering, because I know you said you couldn't do like life course stuff with this, obviously, but like, have you thought a little bit about how that might affect the findings?
Yeah, again, I think if you look at like across the population, so like getting to age 50, there's not a lot of survivorship bias there. But if you start thinking about certain subgroups, there is a lot of survivorship bias. And so we don't have an answer for that. And the aging studies, there's not a lot we can do about it. We can try to kind of triangulate with other studies to see how biased our estimates might be. I think it does mean a lot of our estimates are probably biased towards the null. We all kind of acknowledge that. I also want to point out that like a lot of the exposures that happen to people before they get into a study like the health and retirement study happen in the workplace. So HRS is also trying to understand more those exposures that happened prior to entering the survey. Like I'd like to know about their environments prior to entering the survey, but there are people also interested in occupational exposures. But this is an issue with aging studies. We always start with a group of people who are already slightly selected, although differentially so depending on which population you're thinking about.
Yeah. Question.
I'm not a writer.
We're, two questions have been asked. It's open for faculty.
Thank you. Yeah, this is a wonderful talk. Thank you very much. I'm an assistant professor in geography and the environment. So I'm pretty familiar with the social and physical environment mentioned today, but I'm not familiar with those health-related topics such as epigenetics, like this term. This is my first time to hear about this. I have two questions. The first question is, because as a geographer, I always perform the analysis at neighborhood level, from the broad groups, what you mentioned today. But it seems that those biology-related and health-related topics you mentioned is more focusing on individual level. I'm curious, in your past work, how did you align this? Either like you perform the analysis at the individual level, or you aggregate the data to the neighborhood level and then associate with the neighborhood level environmental factor. That's my first question regarding the spatial unit or the unit that you perform your experiment. The next question is, so I'm also curious, after you examine the associations between the environmental factors and health-related issues, so what's next? We can absolutely observe or quantify the disparity in different neighborhoods. But I'm also curious, like, what could be the next direction? Like, after we have such kind of knowledge, what's next? Any social impacts or actually translate to policy, etc. Because I'm not familiar with this area, so I really appreciate your.
She only has 30 seconds to answer you. Those two questions, so you may have to address these in
a track level database, although sometimes we have it at the block. The geography kind of depends on the question and data limitations. So when I started this work, I could only do census tract, which is not always ideal. Like, and we link it to individual level data, and then we use either we do like a cluster adjustment to get robust data errors, or sometimes we use multi-level modeling, like mixed models approaches if there's a lot of. But we always check the data first if there's enough clustering to require a more complex model, or if we can get away from a model. And then the answer to your next question is, I mean, I wish I could show this slide that I pulled up. I showed at the National Academies workshop, but was a session on environment, midlife exposures. A study had just come out that was done in a couple of neighborhoods in Louisville, Kentucky, where they had greened up. I call it like, it's like a glow up, but it's a green up. They planted trees and bushes and shrubbery. And they checked people's blood-based biomarkers before and after the green up. And we found lower levels of C-reactive protein among the residents in the neighborhood where there was more green things had been added. And that's related to some cardiovascular outcomes like heart attack, which is on the pathway towards dementia. There actually are interventions that can come from this kind of work. They're not as tricky as personalized medicine. They're not necessarily that expensive. I think planting trees and bushes isn't like the most costly intervention we can think of, but they might deliver a lot of bang for their buck. And then you might know that some of the air pollution work that people do, that EPA establishes their guidelines based on the science. And so the science that we do does eventually kind of percolate down into the world programming. So there's a lot of places where neighborhood research is connecting really well with programs. I do think understanding some of the fundamental biology helps us though, and thinking about like, why is this impacting health? Will help us to think about what kind of programs and interventions might really work for people.
Well, unfortunately, I have to cut up the conversation. But this is also a lesson that you should sign up to meet with visitors individually on their schedule and have a chance to meet with them one-on-one. Please join me in thanking Jen.